Good morning. My talk will be about some type of regulatory RNA, which are called the PV-associated RNAs. I will start with a little bit of history. In the beginning of the early days of molecular biology, there was the central dogma which defined the classic roles of DNA, RNA, and protein in eukaryotic cells. And actually, two types of molecules were considered very important, which was first DNA, which was the storage of information, and protein, which was the execution of function. While RNAs were some kind viewed as intermediaries. They served as template, apparatus, adapter for protein synthesis. But with the advent of deep sequencing technologies, it was observed that about 80% of the mammalian genome is transcribed, and that only a small portion of mammalian genome is transcribed in protein coding messenger RNAs. So this indicated that RNAs can have regulatory functions. These RNAs, so besides the protein encoding RNAs, there are non-coding RNAs. And I already mentioned the housekeeping RNAs like transfer RNAs, ribosomal RNAs, but also there are small nuclear RNAs involved in splicing, small nucleolar RNAs, which are involved in the modification and maturation of ribosomal RNA. But besides these housekeeping RNAs, there are more and more indications of regulatory RNAs. And these regulatory RNAs can be in the first division divided into linear RNAs and circular RNAs. Circular RNAs, they are generated by back splicing. So the 3' splice site will splice back to an upstream 5' splice site. So circular molecules are generated, and there is a lot of research now in the regulatory function of these circular RNAs. But we will focus on the linear RNAs, and these can again be divided in two types: the long non-coding RNAs and the small non-coding RNAs. The long non-coding RNAs, they're more than 200 nucleotides in size. They can be as big as messenger RNAs, and they have functions in chromatin modeling and DNA methylation. The small non-coding RNAs, there are three types. First, there are the microRNAs. They're involved in the gene regulation and messenger RNA stability. The small interfering RNAs, they are involved in transposon and virus defense, and this in somatic tissues. And finally, the subject of the talk, there are the PV-associated RNAs, or the piRNAs. They're involved in transposon and virus defense, and typically not in somatic tissues, in germline tissues. And besides silencing, they also are involved in epigenetic regulation. What are PV-associated RNAs? To illustrate this, I show some graphs of sequencing. So in the ordinate, you see the abundance, and in the abscissa you see the length of the small RNAs, typically around 20 to 30 nucleotides. And the first two graphs to the left, you see the sequencing results in somatic cells of a fly, Drosophila, and you see two peaks. So one is around 21 nucleotides. These are the siRNAs, and there is then another broad peak, a little bigger. These are the miRNAs. The two graphs to the right is the sequencing of ovary tissue, which consists of both somatic cells and germline cells. And you see that there is a dramatic difference. So you can see that You see also that there are siRNAs and miRNAs. But there is also, in addition, there is a very broad peak from 24 to more than 30 nucleotides, and these are the piRNAs. Also shown on these graphs are the association of these piRNA, of these small RNAs with protein. So siRNAs are associated with a protein named Argonaute-2, miRNAs with Argonaute-1, while the broad peak of piRNAs are associated with other Argonaute proteins, which are called the PV proteins. This is another example of deep sequencing, and this is on the ovary of human. So the first graph is the sequencing of all small RNAs, and you see that there is a peak around 22 to 23 nucleotides. These are the miRNAs. The graph to the right is the sequencing on a sample which has been oxidized. It was treated with sodium iodide, and sodium iodide will destroy the hydroxyl group of RNA. So these RNAs in which the hydroxyl group is destroyed, they cannot be cloned. So no libraries can be made unless this hydroxyl group is protected. And so in that case, the piRNAs can be sequenced because they have a methylated 3' hydroxyl group. And this is then illustrated in this graph that the piRNAs have a methylated 3' hydroxyl group. So this is a property of the piRNAs, and this is a way to characterize them. And during the sequencing of these small RNAs, one property also became apparent, which is that there is a sequence bias. So the first nucleotide in these small RNAs is a uracil. It's illustrated by the orange color. And then during the sequencing, there is also another feature that appears. The piRNAs, they are from both sense and antisense orientation. But when the sequences were analyzed, it appears that there is a ton nucleotide overlap in the complementary piRNAs, and this is called the ping-pong signature. I will explain this more later. And finally, another properties of the piRNAs is that a lot of them represent transposable elements. What are the PV proteins? PV proteins are a subfamily of Argonaute proteins. They are preferentially expressed in the gonads. They are associated with stem cell function and associated with transposon silencing. They're quite big proteins, 100 kilodalton. They have 4 characteristic domains. And they bind the small RNAs. And what they do is then that they search for targets in the RNA population. They can hybridize to the targets and then trigger silencing, or they trigger degradation. How do the PV proteins acquire the piRNAs? Actually, in the genome of animals, there are genetic loci which are called piRNA clusters. In general, they are located in the heterochromatin. They are consisting of repeat sequences and transposons, and they are transcribed by a dedicated transcription machinery. And typically, such transposon sequences, they always have some transcripts in antisense orientation. After transcription, and these transcripts can be very large, they can be 500 kilobases, they are exported out of the nucleus. This is also a dedicated export machinery, which is different than the export machinery for messenger RNAs. And in the cytoplasm, they are processed by exonuclease called zucchini, which is associated with mitochondria. And this endonuclease activity will generate what is called the precursors of the piRNAs. These precursors of piRNAs then get incorporated in a PV protein and at the 5' end. And because these precursor piRNAs can be quite big, they will be trimmed at the 3' end, and until they fit the size of the PV protein. So this pathway is called the primary piRNA biogenesis, and this typically occurs in somatic cells. In the germline cells, on top of that, in general, there occurs something on top of it. And this is called the ping-pong amplification of the piRNAs. So in the primary piRNA pathway, the PV protein, for instance, aubergine, is associated with a piRNA of transposon sequences in antisense orientation, and it will find a target, which is a transposon messenger RNA, and it will cleave that target. And because the bias of uracil in the primary piRNA, and because the cleavage occurs at position 10, and typically signature is generated of 1U, uracil at position 1 and adenine at position 10. This is a ping-pong signature, and also it explains the 10 nucleotide overlap in the piRNAs. When this cleavage is completed, a new piRNA precursor is generated, and this can then be incorporated in another PV protein, in this case argonauts 3, and this can slice again the piRNA precursors, then new secondary piRNAs of antisense orientations can be generated, and so you can get, this is an amplification mechanism for piRNAs, and the piRNAs can accumulate to high levels, and this typically occurs in the germline cells. Also, this process seems to occur in special organelles in germline cells. So this is, for instance, a nerve cell, which is a germline cell in the follicle of insects. And you can see that the PV protein accumulates as dots around the nucleus. And these special organelles are called nuage in the germline. They can also occur in somatic cells, in particular somatic cells. And they, in that case, they are smaller, but they are called the YB bodies. What are the functions of piRNAs? One of the functions, of course, is transposon silencing. And this, I want to illustrate this with the phenomenon of hybrid dysgenesis. It is observed that in particular strains of flies, when they mate with other strains of flies, and the progeny is sterile. So if this occurs when a female fly of one strain lacks transposons which are present in the genome of the male fly with which the mating occurs, and this is because then the female fly does not have the piRNAs that can neutralize the transposable elements from the male partner. So normally these piRNAs are deposited in the oocyte, and they will provide protection against the transposable elements of the father. And this another function of piRNAs and PV proteins is in the regulation of fertility and the mediation of genetic conflict. This has also been investigated in Drosophila, and specifically in the testes of Drosophila. And this is regulated by a gene pair, a pair of two genetic loci, stellate and suppressor of stellate, which is actually functions like a toxin, antitoxin genetic locus pair, and it functions as a checkpoint for accurate Y chromosome segregation. So on the X chromosome, you have a heterochromatic array of stellate repeats. In the testis, when this is unchecked, it results in very high protein expression, and it forms crystals in the spermatogenic lineage. So, and because of the high expression of these proteins, the spermatocytes will die, and so you will get sterility. On the other hand, on the Y chromosome, there is also a heterochromatin locus with repeat sequences, and which is called the suppressor of stellate. And these locus produces piRNAs, which will neutralize the expression of the stellate protein. So and in that case, normal spermatogenesis can occur. So only in the presence of the Y chromosome you get normal spermatogenesis. Another function of the piRNAs, this is a more extended function, is in the regulation of cellular gene expression. In this case, it's envisioned if piRNAs derived from repeated sequences in the genome can target complementary sequences in messenger RNAs. In that case, piRNAs will work something like miRNAs. miRNAs are small hairpin genes. They are produced by cellular genes. They are processed in the cytoplasm and incorporated in argonaute proteins. And then they scan the messenger RNA population for hybridization and trigger translation inhibition and messenger RNA decay. So the question is then whether piRNAs can work in a similar way as the microRNAs. However, it needs to be considered that the piRNAs are different than microRNAs. First of all, there is the variety. There are many more unique piRNAs than microRNAs. So at least 1,000 times more. And one also has to consider the abundance of individual piRNAs. The piRNAs may be have a large number in bulk, but as individual sequences, their expression can be quite limited. And this is important because you need expression at least of a certain level to achieve repression. And so the question is then if individual piRNAs can have enough high expression level to mediate silencing. Then there is the issue of specificity, whether mismatches will be tolerated or not, will define how many targets can be reached by the piRNAs. And also it needs to be considered that piRNAs may have a different mechanism than miRNAs. The miRNAs, when they have their target hybridized, they recruit complexes, and that recruit a dedicated degradation machinery. While in the case of the PV proteins, it needs to rely on the intrinsic RNAs activity in the PV domain. Nevertheless, there have been quite a number of instances in which it was demonstrated that piRNAs regulate cellular gene expression. And this is one of the best known examples is the sex determination in the silkworm. In the silkworm, the heterogametic sex is the female. It has the ZW chromosome configuration. Like all animal genomes, the silkworm genome has a lot of piRNA clusters that produce piRNAs. And also the W chromosome, it's a very heterochromatic, it has a lot of heterochromatin, and it produces abundant piRNAs. And this was also observed because for one region in the W chromosome, which is the sex-determining region. And it was then found that this sex-determining region encodes a piRNA cluster, which is called the FEM clusters. And it was also demonstrated that a particular single piRNA, which is called the FEM piRNA, was expressed at a reasonably high level, about 200 reads per million. And it was demonstrated that it could cleave a target which encodes a masculinizing factor, a transcription factor encoded on the Z chromosome. So the sex determination mechanism in the silkworm is based on a piRNA produced by the W chromosome that targets a transcription factor that is essential for male development. So, and this has then led to the hypothesis that transposons could contribute to the evolution of genomes by acquiring, because of the acquisition of transposable elements, new piRNA clusters will be generated, new piRNAs will be expressed, and they can then be used to form new regulatorily gene networks. So this fits within the idea that the presence of transposable elements can be a benefit for the evolution of genomes. The piRNAs and the PB proteins, they are specific for the animal lineage. They are not present in plants. You can, on the other hand, you can also find them in a few unicellular eukaryotes like parameciums and ciliates. And it's in generally so far there have been found a variety of additional functions of piRNAs. Some examples are mentioned here, like transgenerational memory in the nematode, regeneration in the flatworm, learning and memory in the sea slug, and so on. But I want to, in the last few minutes, I want to focus on the role that the PB proteins and the piRNAs can have on stem cell functions. And indeed, thank you. And indeed, the first mutant of PB was actually a disruption of stem cell function. And this, and actually, the name PB was derived from this. The abbreviation stands for P element induced with B testis. So what and I want to end with some speculation between the role in transposon sizing and regulation of stem cell function. So with respect to stem cells, there are different types. The most potent stem cells are pluripotent stem cells. And the PB proteins, we can speculate they have protection against transposon activation in the pluripotent stem cells. And this is because of the high plasticity of these pluripotent stem cells, which require an open chromatin structure. And if for their potency, so the pluripotency requires an open chromatin structure, and they are vulnerable for transposon activation. So, and that's why they need expression of these PB proteins. And this is actually observed in animals with a high regeneration capacity, like planaria and hydra, where you have a very high expression of PB proteins in the stem cells. And I want to end with this. And this is my affiliation. I want to mention that we also work with PB proteins in the lab. We look for PB proteins in antiviral defense and the production of viral piRNAs. Thank you.